Hello dear students, welcome back to online biology coaching classes for the preparation of competitive exams specifically for the NEET. So in the previous videos I explained about the complete structure of alimentary canal in the humans and what are the associated glands like salivary gland, gastric glands, intestinal glands, pancreas and liver. And I also explained about the composition of the juices secreted by these glands, what are the different enzymes present in them. And today in this video I am going to explain the process of digestion and absorption. How exactly the carbohydrates are digested, what are the parts of alimentary canal involved in the digestion of carbohydrates, like that the digestion of the proteins, digestion of the fats and the digestion of nucleic acids. So let us move to the details. Digestion, absorption and assimilation of proteins, carbohydrates and fats. How exactly the food is digested in the digestive system, specifically the different parts of alimentary canal. What are the enzymes required for the digestion of the proteins, carbohydrates and fats, and what are their end products where the digested food is absorbed in the alimentary canal. So as you know, the definition of the digestion states that digestion is conversion of complex insoluble food materials into simple and absorbable forms. Whatever the food we consume, it is not directly absorbed by the body cells, so that the complex food material which we consume as our regular diet or as food, first it should be broken down into simple forms, and those simple forms are finally absorbed by the small intestine, particularly the villi region of the small intestine. It absorbs the digested food, so that the absorption is possible only when the complex organic molecules which are present in the food material are broken down into simple and absorbable forms. This process is called digestion. Then how exactly digestion occurs? What are the processes it includes? Whether the digestion is only mechanical process or the digestion is including the chemical process. So it includes both the mechanisms, in that the mechanical processes which are included in the digestion are: the first itself is mastication, carried out by the teeth. It is called chewing. The breakdown of food in the mouth or oral cavity is called mastication. It is also called chewing. Then deglutition means swallowing through the pharynx and esophagus. Then peristalsis, wave-like movement of the food bolus. Already in the mouth the food is mixed with the saliva and the salivary enzymes. Now that partially masticated food, try to concentrate on the terms. The partially masticated food along with the saliva, it is referred as food bolus. And this food bolus, it shows wave-like movement through the gut, that is alimentary canal, by the muscular contraction which is called peristalsis, particularly through the esophagus. So first part of digestion includes mechanical process, and mechanical processes are chewing, swallowing and the wave-like movement of the food bolus called peristalsis. Then there are two basic types in the digestion. One is mechanical digestion, already the process you came to know, and another is chemical digestion. So let us elaborate what exactly happens during the mechanical digestion and how it is different from the chemical digestion. So before continuing, for chemical digestion, prior to this one, means before chemical digestion, the mechanical digestion is compulsorily required, because the food is larger particles of the food are masticated or broken down and it is swallowed. It transferred into the stomach through the mechanical digestion. Then what happens in the chemical digestion? How it is different from mechanical digestion? Let us elaborate. The first one, mechanical digestion. Mechanical digestion, it is initial stage of physical breakdown of the food. Just the food particle or the food molecules are broken down into smaller pieces. The large piece of the food is broken down into smaller piece, and this is carried out by the teeth which are present in the mouth. It occurs mainly in the mouth, where teeth chew the food and tongue manipulates the food. The mixing of saliva with the masticated food is carried out by the tongue. That mixing requires the help of tongue, or tongue helps in the mixing of saliva with partially masticated food. Therefore, the site of mechanical digestion is mouth, where the food particle, the larger food pieces are broken down into smaller pieces. Apart from that, the saliva is also mixed with the food. This is the mechanical digestion. Then what happens in the chemical digestion? Chemical digestion is somewhat different from physical or mechanical digestion. Here, the separation of food into molecular components by chemical means. See, in the regular diet, we consume all the type of the chemical molecules, or I can say nutrients, the better term, nutrients. Our diet plan or our diet includes carbohydrates, we consume protein in different forms, we consume the fat in different forms, and all the plant and animal sources which we consume as the food, they contain the nucleic acids. Therefore, the separation of these components is very much essential step in the digestion, and it is carried out during chemical digestion. Again, the process of chemical digestion also begins in the mouth. So both mechanical digestion and chemical digestion, both of them are initiated in the mouth only, but here the secretion of saliva which contains the digestive enzymes, particularly the salivary amylase or only amylase or taline. Try to remember all the terms. Salivary amylase is carbohydrate digesting enzyme present in the saliva, and salivary amylase is simply known as amylase, or it is also called taline. All these are the same names of salivary amylase. The chemical breakdown of the food continues through the stomach and completed in the small intestine. See how interesting this process is. When larger food pieces are broken down into smaller pieces, this is the mechanical digestion. There will be no involvement of any enzyme. Without the help of enzyme or without involvement of enzyme, the teeth break larger food particles or food pieces into smaller food pieces. It is called the mastication, and it is part of mechanical digestion. But when it comes to chemical digestion, directly the concentration is given for the action of enzymes. That means during the chemical digestion, the separation of the food into molecular components occur by chemical means. Carbohydrates are broken down into glucose, fructose and galactose. Proteins are finally broken down into amino acids. Fats are finally broken down into glycerol and fatty acid. Like that, the nucleic acids are broken down into nucleotides, which further divided into the nucleoside. Like that, the chemical separation occurs during the chemical digestion, and remember, this is completely with the involvement of enzymes. Without involvement of enzymes, mechanical digestion carried out by the teeth. With the involvement of enzymes and chemical separation of the food as molecular components, it is through the chemical digestion. See, the chemical digestion and mechanical digestion, both of them are initiated in the mouth. The chemical digestion continues in the stomach because the stomach secretes gastric juice. Gastric juice contains different enzymes. From that, it is continued in the small intestine because the small intestine secretes intestinal juice. Apart from that, pancreatic juice and bile juice also act on the food in the at the duodenum region of small intestine. Of course, bile juice is the specific juice of alimentary canal secreted by the liver, which do not contain any type of digestive enzymes. Even though the enzymes are absent, but bile juice is required for the digestion of the fat to carry out the emulsification, so that the bile juice is also very important for complete digestion of the fat, and further the complete digestion leads to absorption. So, what are the different parts of alimentary canal showing the chemical digestion? Mouth, stomach and small intestine. So in the large intestine, there are no glands, no enzymes, only there is solidification of the food occurs, absorption of the water occurs. So this is about the chemical digestion. Yes, moving to the physiology of digestion. What we are going to study in this particular part? So that includes digestion of carbohydrates, where it occurs, where it completes, what are the enzymes required for digestion of the carbohydrates. The rice, jawar, wheat, all products of these grains, as well as the vegetables like potato, sweet potato, every food, it is having more or less content of the carbohydrate, which is completely digested. Then digestion of the protein, egg, milk, fish, meat and all the pulses. The pulses are very much Bengal gram, black gram, all these pulses are rich in the proteins. Then digestion of the fat, some fruits, egg, milk, butter, all these products are rich in the fat. And digestion of the nucleic acids, both plant and animal sources which we consume as food, they contain the nucleic acids, even they are also digested. So in the physiology of digestion, you are going to study how exactly the carbohydrates are digested, what is the process of digestion of the proteins, what is the process of digestion of the fats and nucleic acids. And dear students, in this video, I am going to concentrate on two processes. One is digestion of the proteins and digestion of carbohydrates. So you need to remember what are the different enzymes, whether they are secreted in the active form or they are secreted in the inactive form. If they are secreted in the inactive form, how they are converted to active forms. So without wasting time, let us move to the digestion of carbohydrates. The digestion of carbohydrates, it begins or it starts in the mouth or buccal cavity, and it is completed in the small intestine. That means only mouth and small intestine are the parts of alimentary canal which are involved in the digestion of carbohydrates. Then why not stomach? I have skipped that stomach. It is not skipping. In that stomach, the digestion of carbohydrates does not occur, or it is not at all taking place in the stomach. Only the mouth and the small intestine of alimentary canal are the regions of alimentary canal where the digestion of the carbohydrates takes place. Then question is, why not in the stomach? See, the enzyme which acts on the carbohydrate or which breaks the carbohydrate are called carbohydrases. Hope you are getting my point. Any enzyme, if it acts on the carbohydrate, if it breaks the carbohydrate, such enzymes are called carbohydrases. Like that, proteases, enzymes acting on the proteins, or enzymes hydrolyzing the proteins, enzymes breaking the proteins are called proteases. One more term, lipases, enzymes acting on the lipids. Then nucleases, enzymes acting on the nucleic acids. So with respect to this one, there are four types of the enzymes. Carbohydrases acting only on the carbohydrates, proteases acting only on the proteins, lipases acting only on the lipids, and nucleases acting only on the nucleic acids. Why this specification? If you remember, enzymes are substrate specific in their action. The statement I expressed or I told is, enzymes are substrate specific in their action. Then question is, what is a substrate? It is the molecule which is broken by enzyme during any process, and it is called substrate. Enzymes particularly act on related substrates. See, the enzyme protease is present. If carbohydrates are present, there will be no breakdown of carbohydrate, because the proteases can act only on the proteins. If unrelated substrate is available, there will be no reaction or no action of the enzymes. This property is called substrate specific action of the enzymes. So as the gastric juice of stomach does not contain any carbohydrate digesting enzyme, there will be no digestion of carbohydrate in the stomach. Even though some enzymes enter into the stomach, but the acidic environment of the stomach does not allow any carbohydrate digesting enzyme from the saliva which enters the stomach to act on the carbohydrates. So next point, there are two types of carbohydrates in the human food that need to be digested in the alimentary canal of man. They are polysaccharides like starch, glycogen and cellulose. Another group is disaccharides like maltose, sucrose and lactose. What it means? See, in the regular food what we consume, this food contains two types of carbohydrates. One is polysaccharide and another is disaccharide. Both of them are the carbohydrates. Polysaccharide are the complex carbohydrates, disaccharide are the middle range and monosaccharides are the simple range of carbohydrates. When two monosaccharides are joined by the glycosidic bond, that result in the disaccharide. Two sugar components joined by the glycosidic bond. So polysaccharide, complex carbohydrates. The polysaccharides which are present in the regular food of humans include starch will be there, glycogen will be there, cellulose is also there, and even glycogen is stored in the liver itself. The excess of the glucose is converted into glycogen and that glycogen is stored in the liver. And disaccharides include maltose, sucrose and lactose. So all these types of carbohydrates are digested during the digestion process, and partially or some part is digested in the mouth, and complete digestion of the carbohydrate occur in the small intestine. So moving to the next point. Digestion of carbohydrates in the mouth. How exactly the carbohydrate is digested in the mouth and which enzyme is involved in the digestion of carbohydrate, particularly in the buccal cavity, oral cavity or mouth. Saliva, it is the juice secreted from salivary glands. How many pairs are there? 3 pairs of salivary glands: parotid glands, sublingual glands and submaxillary glands. So these glands together represent salivary glands and the secretion is called saliva. The saliva contains a digestive enzyme called salivary amylase. This is the important enzyme to remember. Which enzyme is present in the saliva? The saliva contains carbohydrate digesting enzyme called salivary amylase. See what it does, or what is the function of salivary amylase. This enzyme breaks starch into sugar and pH required is 6.8. And interesting fact is about 30% of the starch is digested by the amylase. Hope you remember, salivary amylase is also simply referred as amylase. Another name of the salivary amylase is ptyalin. Where it is present? The salivary amylase is present in the saliva. And what is that saliva? Saliva is the secretion of salivary glands. On which compound it acts? Salivary amylase acts on the starch, and only 30%, about 30% of the starch is digested by the salivary amylase. And at the same time, you have to remember what is the pH required for the activity of salivary amylase. See, starch which we consume through the regular diet, in the presence of salivary amylase with the required pH 6.8, it is converted into maltose. Then limit dextrins. So maltose, there will be no complete conversion into glucose. The starch is converted into maltose with the help of salivary amylase with the required pH for the action is 6.8. So in the mouth, starch gets converted into maltose in the presence of salivary amylase. So moving to the next point. Another enzyme which is present in the saliva is lysozyme. But this lysozyme, it don't have any activity in the digestion, physiology of digestion. That means it is not acting on any component of the food. Then what is its function? Lysozyme is antibacterial enzyme. Antibacterial, against the bacteria. if living bacteria enters the mouth along with the food in the presence of lysozyme or by the action of lysozyme, that bacteria will be finally killed. So function of lysozyme is to kill the bacteria, therefore it is called antibacterial agent, which is functioning against the bacteria. If any living bacteria enters the mouth along with the food, that living bacteria is killed by the action of the antibacterial enzyme called lysozyme. Next, see, salivary amylase continues to act in the esophagus because esophagus does not contain any gland and there will be no any particular juice secretion, but its action stops in the stomach as the content becomes acidic. Hence, carbohydrate digestion stops in the stomach. Already I told this explanation, but let me repeat. There are two reasons why the carbohydrates are not digested in the stomach. One is absence of carbohydrate digesting enzyme in the stomach. This is one reason. Then second one, even though the salivary amylase it enters the stomach, but in the acidic environment, the salivary amylase stops acting on the starch so that carbohydrate digestion does not occur in the stomach. So it is started in the mouth where starch is converted into maltose with the help of enzyme salivary amylase, with the pH required for the action is 6.8. Only partial conversion occurs because mouth food is kept in the mouth only for short period, so there will be no complete breakdown of the starch. About 30% of the starch is converted into maltose and remaining is as it is transferred into the stomach, and in the stomach there is no action on the starch because salivary amylase is inactivated in the acidic environment and there are no carbohydrate digesting enzymes in the stomach. Then moving to next point, digestion of carbohydrate in the small intestine. What happens in the small intestine? Whether the carbohydrate digestion occurs in the small intestine or not, let us move to the digestion of carbohydrate in the small intestine. The food gets in the small intestine, the food gets mixed with, see, once the food enters into small intestine, here the food from the stomach, now it is entering into small intestine. Already that partially masticated food, swallowed food through the peristalsis movement and by the chemical digestion process, already it has reached the stomach, and now from the stomach the food is moving into small intestine. In the small intestine, the food gets mixed with bile juice as well as it is mixing with the pancreatic juice, it is mixed with the bile juice, and also the food is mixing with the intestinal juice. The three juices are acting on the food. Exactly in which part? In the small intestine. Bile has no enzymes, so has no chemical action on carbohydrates. As the bile juice does not contain any the carbohydrate digestion, not only carbohydrate, see, this is the small intestine, it is duodenum part, and here you can see only part of the stomach. All this is the intestinal portion. Bile juice does not contain any enzyme, not only for the carbohydrate, even protein digesting, lipid digesting enzymes are not present in the bile juice. So there will be no action of bile juice on the carbohydrate digestion. Then moving to Next part, in the small intestine, the pancreatic juice acts on the food. And what happens? Let us move to the details. Action of pancreatic juice on the carbohydrate. Pancreatic juice contains pancreatic amylase. Again the term is amylase. So amylase is the enzyme acting on carbohydrate, which is also called amylopsin. Salivary amylase is also called taline, and pancreatic amylase, which is present in the pancreatic juice, it is called amylopsin. That hydrolyzes polysaccharides, complex carbohydrates, into disaccharides, and the pancreatic enzyme operate in the pH 8.8. It requires alkaline pH, which is provided by bicarbonates present mainly in the bile juice. The salts which are present in the bile juice, they make the pH 8.8, and with this alkaline pH, the pancreatic enzymes act on the food material, though also present in the pancreatic juice. What it means? Bicarbonates present in the bile juice as well as bicarbonates present in the pancreatic juice, they maintain the alkaline pH 8.8. This pH is required for the action of pancreatic amylase, which is also called amylopsin, on the carbohydrate. So what happens? Again, the same reaction, only the pH and enzyme is different. Starch, in the presence of amylopsin or pancreatic amylase, it is converted into maltose. Only the difference is that the pH required is 8.8. So in the previous process or in the previous explanation, starch in the presence of salivary amylase is also converted into maltose. Whatever the starch left unconverted, because in the stomach the conversion of the starch has temporarily stopped, when that unconverted or unbroken or undigested, more specific, undigested starch, when it enters from stomach into the small intestine, in the small intestine, with the action of pancreatic amylase or amylopsin, the starch, whatever the starch which is left as it is finally converted into maltose, and amylopsin requires the pH 8.8. Therefore, conversion of starch, first point, it occurs in the mouth with the help of salivary amylase. pH required is 6.8. What is the product? Maltose. Starch in the small intestine is finally converted. Whichever is left as it is, all that unconverted or unbroken, undigested starch, it is finally converted into maltose by the action of pancreatic amylase. That means in the small intestine, now entire starch is converted into maltose. The next process, what happens to the maltose? How maltose is finally converted? So moving to the last step of carbohydrate digestion. Action of intestinal juice on the carbohydrates. How this intestinal juice helps in the carbohydrate digestion. The intestinal juice, it contains different enzymes which are required for the digestion of carbohydrates. And the pH of intestinal juice, what is another name of intestinal juice? It is succus entericus. Intestinal juice is also alkaline in nature. That is why the pancreatic juice enzymes are acting in the small intestine. Intestinal juice, it contains, these are the terms that you need to remember, very important. 3 carbohydrate digesting enzymes: maltase, sucrase and lactase. See, I am talking only about carbohydrate digestion. Actually, intestinal juice contains the protein digesting enzymes also. But with respect to carbohydrate digestion, I have taken only these 3. In the intestinal juice, 3 carbohydrate digesting enzymes are present. What are those? Maltase, sucrase, lactase. Maltase, the name itself is indicating substrate specific. Previously I explained. Enzymes are substrate specific in their action. So maltase acts or digests the maltose and the end product is glucose. Finally, maltose is converted into glucose, 2 molecules, with the help of maltase. Sucrase is acting on sucrose. This is disaccharide. Maltose, sucrose and lactose are the disaccharides, and finally they are converted into monosaccharides. Already starch, entire starch content which is consumed through the food is converted into maltose. 30% with the action of salivary amylase and remaining is converted with the help of pancreatic amylase, and finally the remaining carbohydrates, maltose is converted into glucose or digested into glucose by the action of maltase. Sucrose is converted into glucose and fructose which is present in the sugar. Sucrose is converted into 1 molecule of glucose and 1 molecule of fructose by the action of enzyme sucrase. Like that, lactose is present in the milk and lactose is converted or digested into 1 molecule of glucose and 1 molecule of galactose by the action of lactase. Therefore, what are the end products of carbohydrate digestion? Glucose, fructose and galactose. The rich source of carbohydrate digestion is glucose, and it is energy rich molecule for the body cells to produce finally energy in the form of ATP. Because when 1 molecule of glucose is broken down aerobically, it produces 38 molecules of ATP. That is why it is energy rich molecule which is belonging to the monosaccharide, which is a type of carbohydrate. Therefore, the carbohydrate digestion started in the mouth, in the stomach there will be no digestion of carbohydrates, and finally the carbohydrate digestion is completed in the small intestine with the help of 3 carbohydrates like maltase, sucrase and lactase. So finally this is the reaction representation. Maltose, in the presence of maltase, it is converted into 2 molecules of glucose plus glucose, 2 molecules. Then lactose is converted into glucose plus galactose in the presence of lactase, 1 molecule of glucose and 1 molecule of galactose. Sucrose is digested in the presence of sucrase. The resultant products are 1 molecule of glucose and 1 molecule of fructose. It is fruit sugar. So all these are the products of complete digestion of the carbohydrates which are absorbed in the small intestine, and the digestion of the carbohydrates is completed in the small intestine itself. The carbohydrate digestion, how many enzymes you came across? Only 5. Salivary amylase of the saliva, pancreatic amylase or amylopsin of the pancreatic juice, maltase, lactase, sucrase. These three are the enzymes present in the intestinal juice. So both salivary amylase and pancreatic amylase are acting on the starch and it is converted into maltose. Maltose is converted into two molecules of glucose by the action of maltase. Lactose is digested into glucose and galactose, one molecule of glucose and one molecule of galactose in the presence of lactase. Sucrose is digested into one molecule of glucose and one molecule of fructose in the presence of sucrase. So final digestion of the carbohydrates and complete absorption of the carbohydrates, both of them are occurring in the small intestine. So this is about the complete digestion of carbohydrates. So moving to the next one, digestion of proteins. It is opposite to that of carbohydrate digestion, because already you have studied the carbohydrate digestion, where it starts in the mouth and where it is completed in the stomach. But here the digestion of proteins begins in the stomach. That means there is no digestion of protein or breakdown of the protein in the mouth or buccal cavity. Simple, because the saliva secreted from the salivary glands does not contain any proteolytic enzyme or protein digesting enzyme. Therefore, the digestion of protein, it begins in the stomach and it is completed in the small intestine. See, for ultimate process of digestion, small intestine is the last destiny for the absorption as well as for the digestion, whether it is carbohydrate, whether it is protein, whether it is fat or it is nucleic acid. All the molecules are finally completely digested in the small intestine only, and they are absorbed in the small intestine itself. So stomach and small intestine are the parts of alimentary canal which are the sites of protein digestion. The enzymes that act on proteins are known as proteases, or they are also called peptidases. Either they are called proteases. Proteases is the very common term used for the enzymes acting on the proteins, are also known as peptidases. So protein digestion is confined or it is restricted to stomach and small intestine. It begins in the stomach and it is completed in the small intestine. But let me tell you, see, only with the help of 5 enzymes, the carbohydrates are completely digested. But protein digestion requires many enzymes which are present in the pancreatic juice, which are present in the gastric juice, and which are present in the intestinal juice. Set of enzymes which are required for protein digestion are more than that of carbohydrate digestion. So let us know what are the different enzymes required for protein digestion. Digestion of proteins in the stomach. What happens first in the stomach? Semi-digested food that rests in the stomach for 4 to 5 hours. Once the swallowed food reaches the stomach, that food remains in the stomach for about 4 to 5 hours. Then gets mixed with the acidic gastric juice by churning movement of the muscular wall of the stomach. Slowly the gastric juice is mixed with the food, and now that mixture is called chyme. The swallowed partially masticated food or the bolus which has mixed with the saliva, now it has reached the stomach, and in the stomach it is mixing with the acidic gastric juices, and this mixture it is referred as chyme. Chyme is semi-fluid, it is not completely solidified structure, semi-fluid mass into which food is converted by gastric secretion and which passes from stomach into the small intestine. Gastric juice contains, this is important, already you know about this one. What are the components of gastric juice? It contains HCl. Then question is, why the HCl is important, how it plays important role in digestion, that we will come to know. HCl, pepsinogen, renin and lipase. Pepsinogen, prorenin inactive form and lipase, gastric lipase are the enzymes which are present in the gastric juice. Apart from that, the acidic nature of the gastric juice is because of HCl. Hydrochloric acid dissolves bits of food and creates an acidic medium so that pepsinogen is converted into pepsin. Pepsin is a protein digesting enzyme. It is secreted in its inactive form called pepsinogen, which then gets activated by hydrochloric acid. HCl provides optimum pH of 1.2 to 1.8 for the pepsin. What it means? The digestive enzymes which are present in the gastric juice are pepsinogen. Another is renin. Actually it is secreted as prorenin. And one more is gastric lipase. Pepsin and renin, both of them are secreted in the inactive form. The inactive form of pepsin is called pepsinogen. Please try to remember these terms. Pepsin is secreted in the inactive form and inactive form of the pepsin is called pepsinogen. To convert inactive pepsinogen into active pepsin, HCl is required. This is how the HCl is important for the digestion. Inactive pepsinogen is converted into active pepsin in the presence of HCl. And what is the pH of gastric juice? 1.2 to 1.8, highly acidic. And which component of the gastric juice is responsible for the acidic property? HCl is responsible for the acidic property of gastric juice and it is 1.2 to 1.8. So thing to remember is, the enzymes of the gastric juice are secreted in the inactive form. For the conversion, it requires HCl. Pepsinogen is converted into pepsin in the presence of HCl. Pepsinogen, it is the indication of inactive enzyme, and inactive enzymes are called chymogens. Already I repeated this term in the previous videos also. The pepsin is active form, the inactive form is pepsinogen. Pepsinogen is converted into pepsin by the action of HCl. Then here is the conversion. Pepsinogen is converted into pepsin in the presence of HCl, and once the pepsin is activated, it acts on the proteins. In the presence of pepsin, the proteins are converted or broken into simple forms like proteases and peptides. When pepsin acts on the proteins, the protein, complex protein is converted to simple forms like proteases and peptides. Further, they need to be conversion or they are required to convert. Then prorenin, inactive form of renin. Even renin is also secreted in the inactive form. The inactive form of the renin is called prorenin. In the presence of HCl, the prorenin is converted into renin, and renin is acting on milk protein called casein, and casein is converted into para-casein. The milk protein acting enzyme, very specific. Renin acts on milk protein called casein, and this casein is converted into calcium para-caseinate. Actually, the calcium is required for the action. Further, I will show the reaction. Renin plays an important role in the coagulation of milk. That means solidification of the milk is possible by the action of renin. This is how the milk is coagulated. Yes, look at the reactions here. Prorenin, which is inactive form, it is converted into active renin by the action of HCl. Once the renin is activated, it acts on the milk protein called casein, and casein is converted into para-casein. This para-casein, in the presence of calcium ions, it gets transformed into calcium para-caseinate. It is nothing but coagulation of milk. The solidified form of the milk is nothing but a curd, and that is possible with the help of para-casein. And para-casein is converted from a form of the casein with the action of renin. Finally, the calcium para-caseinate, in the presence of pepsin, it is converted or digested into peptones. Now this digestion is completed. So in the stomach, on the proteins, only two enzymes are acting. One is pepsin and another is renin. And both of them are secreted in the inactive form. Pepsin is secreted as pepsinogen and renin is secreted as prorenin. The conversion of inactive enzymes into active forms is carried out by the HCl. Then finally, the renin is very much essential to act on the milk protein called casein, which is converted into para-casein. In the presence of calcium, the para-casein is converted into calcium para-caseinate, which is the coagulation of the milk, and calcium para-caseinate, it is further digested by the pepsin into peptones. This is how the digestion of the proteins occur in the stomach. Only first part I explained. Still, you need to know what happens in the small intestine, how proteins are finally digested in the small intestine. The food in the stomach is thick, it is acidic and semi-digested. It is called chyme. Need to remember the definition. The mixture of the food, the semi-solid or semi-fluid content of the food, which is quite thick and acidic due to the mixing of HCl, semi-digested, partially digested food which is present in the stomach with thick and acidic property, it is called chyme. So now the digestion of proteins in the small intestine. See, when we consider small intestine, the two types of enzymes are acting. One is the own intestinal enzymes and other is of the pancreatic enzymes, because the food does not enter into the pancreas as well as liver, so that pancreatic juice as well as bile juice, both of them are acting on the food at the duodenum itself. So in the small intestine, what happens to the proteins? When chyme is transferred to the small intestine, it is acted by three enzymes present in the small intestine. Pancreatic juice with the pH 8.8, 8.3, both of them are correct. Intestinal juice, which is known as succus entericus, and bile juice with the pH 8. Pancreatic juice and bile reach the duodenum through hepatopancreatic duct. This also you need to remember. Which duct is transferring the bile juice and pancreatic juice? It is hepatopancreatic duct. So first, let us know what are the different enzymes of the small intestinal juice that are acting on the proteins. Okay. Trypsinogen is present in inactive form in the pancreatic juice. The enzyme enterokinase, also called enteropeptidase, secreted by the intestinal mucosa, that means it is present in the intestinal juice, activates trypsinogen into trypsin. Again, trypsinogen. Pepsinogen, what I told for pepsinogen, it is inactive form. Like that, trypsinogen is also inactive form of the enzyme which is present in the pancreatic juice. For the conversion of trypsinogen, the required enzyme is enterokinase, where it is present. Enterokinase is present in the intestinal juice, and enterokinase is also known as enteropeptidase. Inactive form of trypsinogen in the presence of enterokinase, it is converted into active form called trypsin. Two more enzymes are present in the pancreatic juice for the digestion of the proteins. Of course, they are also in the inactive form. Chymotrypsinogen, in the presence of trypsin, it is converted into chymotrypsin. Once the trypsin is activated, it helps in the conversion of other inactive enzymes, or it participates in the conversion of other inactive enzymes. Procarboxypeptidase, it is also inactive. Prorenin, like that, procarboxypeptidase. So prefix and suffix both you remember. If gen is present along with the enzyme name, definitely it is inactive. If pro is added for the enzyme's name, it is also inactive form. Pepsinogen, then trypsinogen, chymotrypsinogen, procarboxypeptidase, prorenin. So procarboxypeptidase is also inactive form, and in the presence of trypsin, it is converted into carboxypeptidase. So these three enzymes, trypsin, chymotrypsin and carboxypeptidase, these three enzymes are present in the pancreatic juice. Let me mark this one. Pancreatic juice contains trypsin, chymotrypsin as well as carboxypeptidase. All the three are required for digestion of proteins. Already you have studied about two of the gastric juice. What are those? Pepsin and renin. Now three more enzymes are added: trypsin, chymotrypsin and carboxypeptidase. But all these enzymes are secreted in the inactive form. Gastric enzymes are activated with the help of HCl. Trypsinogen is activated with the help of enterokinase, which is also an enzyme but present in the intestinal juice, also known as enteropeptide. So chymotrypsinogen and procarboxypeptidase are activated by the action of trypsin. So coming to the last bit of the protein digestion explanation, what happens? Finally, once the trypsin, chymotrypsin and carboxypeptidase are activated, trypsinogen is activated by enterokinase, chymotrypsinogen and procarboxypeptidase are activated by trypsin. They act on the proteins which are not digested in the stomach, peptones and proteoses, and finally they are converted into dipeptides. So carboxypeptidase, what is this? This is also the enzyme present in the pancreatic juice. Carboxypeptidase, it acts on the carboxyl end of peptide chain and help in the releasing of last amino acid, because amino acids contain both the groups, carboxylic group as well as amino group. That carboxylic end, it is released by the carboxypeptidase of the last amino acid. This is how the pancreatic proteases, they act on the proteins, and finally peptides with the help of carboxypeptidases, they are converted into small peptide chains and plus some free amino acids are released. Then finally in the intestine, what are the protein digesting enzymes present in the intestinal juice? Intestinal juice, it is also called succus entericus. It contains enterokinase, but its function is not to break down any molecule, not to break or not to act on any chemical compound. Its function is to activate inactive trypsinogen into active trypsin. And it contains two protein digesting enzymes, aminopeptidases and dipeptidases. Function of enterokinase already you came to know. Enterokinase converts trypsinogen of pancreatic juice into trypsin. Then these two protein digesting enzymes, aminopeptidases and dipeptidases, what they do? The large peptides are acted by or digested by aminopeptidases, which is converted into dipeptides and amino acids. Finally, the dipeptides are digested by dipeptidases to release the free amino acids which are absorbed in the small intestine. This is how the protein digestion is completed in the small intestine. It begins in the stomach and it is completed in the small intestine. So totally how many enzymes are required? Pepsin, inactive form is pepsinogen. Renin, inactive form is prorenin. Conversion is carried out by the action of HCl. Pepsin and renin are the enzymes present in the gastric juice. Trypsin, secreted as a trypsinogen, activation is carried out by the enterokinase of the intestinal juice. The two more enzymes, carboxypeptidase, procarboxypeptidase is inactive form, chymotrypsinogen is also inactive form. These inactive forms are converted into active forms such as chymotrypsin and carboxypeptidase by the action of trypsin. Intestinal juice contains two protein digesting enzymes, aminopeptidases and dipeptidases. This is how finally the proteins are broken down into, digested into amino acids, which are the end products of protein digestion, and these amino acids are absorbed in the small intestine. So this is about digestion of carbohydrates and proteins. Hope this explanation is clear for you. Thank you.